Hello students, welcome to my class. Today we are going to study a small part from elective paper BOD 102, that is Research Methodology, Bioinformatics and Biostatistics. This is a small module from Unit 1, that is Basic Concept of Research and General Laboratory Practices. Module name is Access to Laboratory. Module number is 4. Myself, Dr. Ranjita Savaikar, Associate Professor, Department of Botany, PSS RSN College, Ponda. This is going to be the outline of my presentation. We are going to define what is laboratory, access to laboratory, personal safety in laboratory, good practices to be followed when we are working in laboratory, and the importance of laboratory cleanliness. After I complete this module, you will be able to explain importance of laboratory. You will be able to cite methods of personal safety in laboratory, and you will be able to describe the good practices and rules of laboratory cleanliness. Now, for the first time, when you enter a research laboratory may seem like a maze with numerous things to learn, safety procedures to follow, rules to obey, hazards to contend with, new equipments to use, etc. A laboratory is a place equipped for scientific experiments, research, testing, analysis, and observation. Now, in a laboratory, there may be different types of microbes, plants, animals, chemicals, instruments that are used for experimentation. This is a typical setup of a laboratory. You can see the setup of chemistry laboratory, zoology and botany laboratory. And in a laboratory, besides the instruments, there are museum specimens too. In this picture, you can see the different instruments as well as the experimental setups. Here, you can see different types of laboratory instruments. In botany laboratory, we use oven, centrifuge, we use laminar airflow, BOD incubator, autoclave, hot plate, etc. This is the arrangement of some reagents in a laboratory. Now let us understand access to laboratory. Here we are going to see who should be entering the lab, how you should be working inside the laboratory. Only authorized persons should have access into the laboratory. Children should not be permitted to enter the lab for one simple reason, because there are different types of chemicals, different types of plant specimens, microbes, etc. All the laboratories in which high-risk organisms or chemicals are handled should display the international hazard symbols. Animals, other than experimental animals, should not be allowed to enter inside the laboratory. Laboratory inventory should be maintained to know where and how much hazardous chemicals are there in the laboratory. All laboratory personnel should undergo training to know the security procedures. This is very important. While working in the laboratory, researchers are exposed to different kinds of laboratory hazards like chemical hazards, fire hazards, biological hazards, radioactive hazards, noise hazards, electrical hazards, which can cause damage to the health of the researchers. We need to take utmost care while working in the laboratory. So let us see some of the measures of personal safety. Researchers, while working in the research laboratory, should take care and they should wear a typical type of clothing. That is, the lab coats. There should be a face masks, uniforms, Then the researchers, if they are handling the blood or microbes or infected plants, animals, poisonous plants, hazardous chemicals, it is appropriate to wear the gloves. This picture shows the researchers doing work in the research laboratory with the protection gears. Protection of face. It is always better to use goggles and face shields for the protection of eyes, face from the splashes and radiations. As far as footwear is concerned, it is not advisable to use open-toed footwear in the laboratory. Food consumption. Eating, drinking, smoking, and applying cosmetics or storage of food meant for human consumption should be strictly avoided in the laboratory. Researchers should not run the experiment unattended. Whenever you are doing some experiments, you should always be there. Do not run it unattended. Minimum flammable substances should be stored in the laboratory. While doing experiments, the researcher needs to pipette out the solutions. Pipetting any solution or water by mouth should be strictly avoided. Aspirator bulb can be used for pipetting out the solutions. We need to stick labels on several bottles or test tubes. Leaking of the labels should never be done in the laboratory. Spillage of chemicals, breakage of glassware, leakage of gas should be immediately reported to the laboratory staff. Appropriate procedure for cleaning any spillage or breakage should be laid down and followed. Any material like documents, books, notebooks that is to be taken out of the lab must be protected from contamination while it is inside the laboratory. Safety systems for protection against fire, electrical emergencies, or any other accident should be readily available. Every lab should have the first aid facility that can be easily accessed. There should be sufficient and separate storage spaces for the solutions, reagents, and specimens. Wash basins with running water and detergent should be available. The entrance and the exit of the laboratory should be designed in such a way that it can be easily evacuated in case of emergency. Let us understand the laboratory cleanliness. Wherever we are doing research, we have to maintain that laboratory very neat and clean. Entire lab should be kept clean and free of unwanted materials. Workbench tops should be decontaminated at the end of the day. All contaminated materials like specimens and cultures must be decontaminated before being disposed and cleaned for the reuse. What are the good practices? A researcher must respect the dangers of high voltage, ultraviolet light, heat sources, and cryogenic materials. When transporting hazardous substances, make sure that the containers are sealed. Try not to take routine manipulations for granted, especially those involving glass, needles, or sharps. If the researcher is tired or distracted, he or she must take a break, must relax for some time, and then refocus. If any unsafe conditions are noticed or an accident or injury, you must always talk to the supervisor. There should not be overcrowding of equipments in the laboratory. For specific type of works, appropriate and special attention should be paid to all matters relating to safety problems. it is important to remember and comply with the safety instructions. These are the references for additional reading. Thank you.